Welcome, and thanks for spending your afternoon with us. This is Pat Furlong from PPMD, and I'm happy to say that today's discussion for this webinar is on Domagrozumab. It will be referred to as DOMA, and I'm happy to have Dr. Lawrence Charnas with us and Shannon Marafino. We've worked with Dr. Charnas and Shannon for many years, and really happy to hear about the DOMA study and what they've learned throughout. So with that, I'm going to turn over the webinar to Shannon and Dr. Charnas.
Thank you, Pat. Before I get started, I just wanted to thank you for the opportunity this afternoon to review the Domagrozumab study with you. And just before we get started with that review, I wanted to just take a moment to reflect on my own journey with the Duchenne community, which started back in 2011 when Pat came to Pfizer and introduced us to a family, a mom, a dad, and a son with Duchenne. The family had recently lost another son to DMD, and despite that recent loss, they courageously came to Pfizer to tell their story and assure that we understood the urgent need for effective therapies to help these boys. Later that year, we began to design the clinical development program for Domagrozumab, and it was around that time that I attended my first PPMD Connect conference, which was small enough at that time that we actually got up and introduced ourselves. From these early interactions, I quickly learned about the DMD community that included patients, their families, healthcare providers, researchers, and industry partners, all with steadfast and passionate commitment to these boys. As we worked on the clinical trial design, we were faced with many challenges, such as what endpoints could we use to measure a treatment effect of an anti-myostatin therapy, and are they important to patients? Also, how can we assure that when the study is complete, we will have a definitive answer on the safety and effectiveness of this drug for these patients who we know are urgently waiting for new therapies? During this challenging time, we repeatedly reached out to this community to help us navigate our way. So while there are two of us today, Lawrence and myself, who will be providing you with this update, it has taken a village to get us to this point, and for that, we are grateful to this community.
Thank you, Shannon.
So I just wanted to let you know that this information that is being provided today is publicly available for you, and it's being shared in a response to PPMD's request to communicate these study results. And just an overview of today's presentation. We'll be talking about the background of Domagrozumab, what our study design was. Then we'll jump into what the results were for this trial, the demographics, that's who we had enrolled in this trial. I'll tell you about the pharmacokinetics and the pharmacodynamics, which I'll explain more when we get to that section. And then Lawrence will review with you the safety information, focusing on left ventricular ejection fraction, glutamate dehydrogenase, which we refer to as GLDH, cardiac troponin I, and then I'll walk you through the efficacy data, the imaging, and then our plans for data reporting and data sharing. So Domagrozumab was potentially a treatment for Duchenne muscular dystrophy. Myostatin, or growth differentiation factor 8, is a protein that's produced by muscle cells. Its role is to regulate muscle growth and to promote the formation of fibrotic tissue to replace damaged myofibers. Domagrozumab is a neutralizing monoclonal antibody that is a myostatin blocking action. In the MDX mouse model, myostatin inhibition reduces fibrosis and fatty infiltration, and additionally, muscle mass and strength were increased. In the cynomolgus monkey, there was a dose-dependent increase in muscle mass observed. With that information, clinical trials were initiated to evaluate if Domagrozumab could potentially slow disease progression in Duchenne patients regardless of their genotype. In August of 2018, a decision was made to terminate the two ongoing trials in DMD. This decision was based on the phase 2 study not meeting its primary efficacy endpoint, which was to observe a mean change in the fourth stair climb time in patients treated with Domagrozumab for 1 year versus placebo. And in addition, we also evaluated the totality of evidence, including secondary endpoints that didn't support a significant treatment effect either. And some of those secondary endpoints included looking at the changes on 6-minute walk distance, the North Star Ambulatory Assessment, the 10-meter run walk, the performance of upper limbs, and forced vital capacity, which measures respiratory function. So these studies were not terminated for safety. And since the announcement in August, we began to ask sites to perform early termination visits according to our protocol, and those visits were completed at the end of November in 2018. Following that, we finished cleaning our data and locking our database. These steps were necessary to ensure that we had high-quality data when the study was finally complete and prepared for the final analysis and reporting of the data. We are now in the process of completing that reporting, and this process will be fully complete in June of this year. So the publication results plans are to share this data on clinicaltrials.gov as well as in Europe on the Clinical Trials Registry. Pfizer.com also provides a clinical trial synopsis of those study results, and we will plan to publish data as well in scientific journals. So despite the phase 2 studies not meeting their primary objective, the data collected from these studies is of high quality and will support the development of new therapies for DMD. So this was our phase 2 protocol design. We enrolled boys into the study who were 6 to less than 16 years old. And at the time of screening, they needed to be able to perform the 4-stair climb in 2.5, but not more than 12 seconds. They had to be on a background of glucocorticosteroids for 6 months. And once they were enrolled into the trial, they were assigned into one of 3 sequence groups. Sequence groups 1 and 2 received active drugs during year 1 in a dose-escalating fashion. Every month, they would come in for a dose, and for 4 months, they would receive each of the dose levels. So after they completed the 5 mg per kg dose, they were dose escalated up to 20 mg. They received 4 doses up to 20 mg, and then they were dose escalated up to the 40 mg per kg dose. Patients who were assigned to sequence group 3 received placebo all the way through that first 48 weeks. Sequence group 1 went on in the second period of the study to continue to receive the high dose of the study drug. The sequence group 2 patients received placebo in the second year, and in sequence group 3, patients went on to receive the active drug. Once patients completed the 2 years of the study, they were then offered the opportunity to go on and join the open-label extension study. So at the time of our primary completion, we had 120 boys who had completed through 1 year of the trial, and we had about 60 patients who were still remaining in period 2, and about 60 who had gone on to the open-label extension. So our study enrollment on our phase 2 study started in November of 2014, and our open-label extension started about 2 years later, which makes sense, and that's about 2 years when the first boys had completed the trial. We had 8 countries who participated in the study, with 31 sites participating. We screened 162 boys, and we enrolled 120 into the trial. 7 were discontinued during the trial, 65 completed, and 48 were terminated early when we announced the termination of the study. The primary reason that the boys' screen failed was due to not meeting the 4-stair climb time criteria. We also, for the 7 boys who discontinued, 4 no longer wished to participate, 2 decided for other reasons to no longer continue, and one patient had a serious adverse event of worsening anxiety and determined to discontinue the study. In the open-label extension study, we screened 61 boys, and we enrolled in dose 59. 4 were discontinued, and 55 were terminated early. And the screen failures were primarily due to the termination of the study prior to them getting into enrollment. So 4 discontinued from this study, 2 no longer wish to participate, 1 died following a tibia fracture that resulted in a fat embolism, and 1 participant moved away from the site during the ongoing trial. So one of the complexities that we had to consider when enrolling this type of trial was how to design the study to evaluate the effectiveness of Domagrozumab when we were testing various dose levels of this drug. And in order to consider that information, we decided to consider sequence group 1 and 2 as a single active treatment arm as compared to placebo. So when you see the efficacy data for the trial, you'll see us compare the 80 Domagrozumab treated patients against the 40 placebo. However, we were still able to evaluate the safety within each dose level separately. We also wanted to consider the data beyond period 1 and through period 2, and specifically to see if there were any potentially long-term treatment effects with Domagrozumab. As we have seen in other Duchenne studies, that it can be difficult to see changes on some of these endpoints when you're only following this population for a year. So we had the opportunity in this study to look at patients who received 2 years of treatment and to compare them back with a matched natural history control data set. And for the purposes of this study, we were able to have access to the Synergy data set, which is a cooperative international neuromuscular research group. This is a collection of data from 440 Duchenne patients aged 2 to 28 that were followed for up to 10 years. So from that data set, we were able to predetermine a matched control based on their age, their forced stair climb time, and their background of glucocorticosteroids to compare them against our sequence group 1 who received drugs for 2 years. So the first thing we always want to look at when we evaluate data is if our treatment groups were balanced, or did we look at patients who were similar when we're looking for a treatment effect across both groups. So what we saw was the age distribution in the study. Primarily, the boys who were enrolled were around between 7 and 10 years old. However, the mean age was relatively similar, with the placebo being perhaps slightly older, and we also saw that the weight in the placebo boys was slightly higher. And that was driven by 2 patients in the placebo group who drove that weight to be slightly higher. But in general, we would say that those groups were similar. Although age is important, what we learned when we looked at the natural history data for the study, that age may not be as much of a predictor of how the patients would perform on the forced stair climb as compared to what their forced stair climb time was at the baseline when they came in at screening. So we did not do a sub-analysis, or we don't plan to do a sub-analysis where we look for differences by the age of the subjects when they came into the trial, but rather we planned a sub-analysis where we looked at patients who came into the trial and what their forced stair climb time was. And what we had proposed was that there might be 3 different groups enrolling in this trial. One group who may be less likely to decline over 1 year, and we thought that those patients might have had a forced stair climb time that was less than 3.5 seconds. And those who may be in a more rapid phase of decline, boys who perhaps had a forced stair climb time of 3.5 to less than 8 seconds. And then boys who might lose their ability to climb the forced stairs over 1 year, who may have had a forced stair climb time of over 8 seconds. And unfortunately, we didn't see any differences when we looked at those subgroup analysis. So one of the important aspects of doing any clinical trial is to consider the pharmacokinetics and pharmacodynamics. So when we think about pharmacokinetics, it's really what the body is doing to the drug or how the drug moves through the body once it's infused. So in our study, we measured the PK in the blood. So we were measuring Domagrozumab in the blood, and we found that the concentration of Domagrozumab in the blood increased as predicted when we increased the dose of drugs over time. We also want to understand how to account for the drug's interaction with the body, and that's called the pharmacodynamics. And in particular, we wanted to know how the drug was interacting with GDF8 or myostatin. So if you remember, the purpose of the anti-myostatin is to block the action of myostatin. And so we wanted to be able to measure if the drug was doing that. And what we found is that of the available myostatin that was circulating in the blood, about 90% of it was targeted and blocked by Domagrozumab. And now Lawrence is going to go through the safety information with you.
Thank you, Shannon. I'm going to talk a little bit about some of the safety findings with this study. In general, Domagrozumab was safe and well tolerated. This slide presents a summary of the adverse events in the different sequence groups and in total. And safety is reviewed looking at reported events and also sometimes with laboratory findings. That becomes important as you'll hear. When a new drug is put into patients, one can anticipate what might happen, but one has to look at all of the events and all of the data and exercise a little bit of clinical judgment. So we use adverse events as our guideposts to understand safety, and we compare the frequency of events and the severity of events in the different sequences to see if they are similar or if they are different. Using that criterion, the event number and the event seriousness and the event severity was very similar across all three groups, leading us to conclude that Domagrozumab, from an adverse event perspective, was safe and well tolerated. You've heard that there was one individual discontinued in the first year because of anxiety. There were serious adverse events as well in both the initial study and in the open label extension. They were all adjudicated by an internal committee at Pfizer, and none were considered related to the drug and were considered to be events that would happen, as you've heard. More specifically, after we moved from the collection of reported adverse events, we looked at some of the laboratory data where we had specific targets of concern. One was to look at the left ventricular ejection fraction. This is a measure of how well the heart pumps blood from the left ventricle. A normal value is greater than 55%, and if there's a dramatic change in that number, that could be reported as an adverse event. But we were also collecting information using either cardiac MRI or echocardiogram to look at the amount of change from baseline in boys over the course of the study. What is known is that left ventricular ejection fraction declines over time. The rate is estimated somewhere between 1% and 3% annually, but multi-center clinical trials like this have not been done in Duchenne, so we're assessing this for the first time. And we had identified what appeared to be a numerical difference between the sequence groups at week 97, and I will show you that data later. As I had mentioned, safety in an investigational new drug has a lot of clinical judgment applied to it. And the goal with limited data is to look at all the information, identify if a signal is detected or is present, and then identify whether there is a risk to the patients who are receiving the drug. And there are formal processes to do that, and this is listed in the slide with the Pharmacovigilance and External Data Management Committee. And there was also classifications of risk that are using language from regulatory authorities. Basically, the three categories are no risk, maybe there's a risk, which is called potential, and observed risk, which means, yes, there's something seen. We had looked at this change in the left ventricular ejection fraction, went through the analysis, and considered this as a potential, and a letter was issued to investigators and regulators as part of our process of making sure that safety of the patient is paramount. However, we did not recommend changes to the protocol, and had the protocol continued and was not terminated early, we would not have changed anything except to increase the frequency of monitoring to make sure that we understood what the risk was if it were in fact a real. I'll show you the data to explain that now. At the end of week 49, you can look at the changes from baseline in the left ventricular ejection fraction. This particular plot is called the box and whiskers, and in sequence 1 on the left, these are individuals who will continue throughout years 1 and 2 on Domagrozumab. Sequence 2 had the same exposure in year 1 as sequence 1 did, and sequence 3 was the placebo. You see that there is no change from baseline in sequence 3. The mean is around 0. In sequence 2, it's around 1% or so, and that's that cost bar. And at sequence 1 was about 2% change from baseline. These are all within the estimates of previous reports, and that looked fine. And was consistent with our prior knowledge. When we look at week 97, the first thing to notice is that we had much less data. So instead of having roughly 40 boys in each group, we had 23 or 21 in each group, so we had half as much data. And I think what you can see is in sequence 3, the change from baseline is still around 1% or around 0. In sequence 2, it's a little greater, minus 1.6. But there is an apparent numerical difference, not statistically significant, in sequence 1, which is about 4%. And it were these data that led us to do the formal analysis and determine that there was a potential risk. In addition to looking at left ventricular ejection fraction, we also looked at cardiac troponin. Cardiac troponin is a muscle used to help the heart contract. And if the muscle membrane or heart membrane is leaking, this protein will leak into the blood and can be measured. In routine clinical care, cardiac troponin is used as a marker of injury to the heart muscle in myocardial ischemia or a heart attack. It has been reported to be elevated in boys with Duchenne, but the clinical relationship to events is a lot less clear than in individuals who have no risk from muscle disease. There's a lot of data here. The summary is that many of the boys had elevations that were not sustained. They were not associated with clinical symptoms. I think if you look at the baseline, many were above the normal range from the beginning, about 20%. And this fluctuated throughout the study, but the individuals who were elevated at baseline were not necessarily elevated later on, which the reason would say they were not sustained. So this kind of variability is seen What it means is not particularly clear, but this was a rich data set to understand cardiac health and how these measurements move in boys with Duchenne in a controlled clinical trial. We also looked at a novel marker for liver injury. This is an enzyme called Glutamate Dehydrogenase, or GLDH. The rationale for this in Duchenne is that the typical measures of liver health are enzymes abbreviated ALT and AST that are released when a liver cell is injured. However, these enzymes are also elevated in boys with Duchenne, and it's very difficult to detect a subtle signal. So a liver-specific marker that would not be influenced by leaky muscles would have a lot of value in the field of Duchenne. As we noted here, GLDH was evaluated. It's largely a liver-specific enzyme. There's a bit in muscle, but not enough to make a significant rise. And it has not been used previously in Duchenne. It has been shown in animals to be quite specific for liver. And on the next slide, what you'll see is the results from serial studies for boys over the course of the study. We have measurements roughly every 4 weeks of all boys in the study, so these are close to 2,000 values. The sequences are color-coded, and I think what you can see is that virtually all of the results are below the normal limit. That's 1, that's where the line is. And if you go to 2, which is the general threshold for when you expect to see liver injury of any sort, this could be from a virus. Children, that's the most common reason. You see a few data points that rise, and they're generally, in this case, more are red than others, but they were not sustained. And we conclude from this that there's not liver injury from Domagrozumab, and that the biomarker is very good in boys with Duchenne. I'm going to turn it back to Shannon.
Thank you. So now I'm going to walk through with you the efficacy endpoints of the study. And as I said, the primary endpoint for the study was not met, which was mean change from baseline on the forced stair climb time. In addition to the primary analysis, we also conducted additional sensitivity analysis on the forced stair climb. So we did see some small favorable directionality, but there was no statistically significant treatment effect that was seen. We also did a comparison following 2 years of Domagrozumab with historical control, and we didn't see any differences on the forced stair climb time. For the North Star total score, we didn't see any statistically significant differences at week 49. However, we did see small directionally favorable difference with the treatment. The 6-minute walk distance, the forced vital capacity, the performance of upper limbs, the time to stand, and the 10-meter run walk also showed no directionally favorable differences compared to placebo at 49 weeks. And then finally, we also included in our study the Pediatric Outcomes Data Collection Instrument, which is a questionnaire that's completed by parent proxy that asks questions about activities of daily living, such as combing your hair, opening doors, getting out of bed, as well as children's willingness or ability to play in other activities and groups. And we didn't see any statistically significant differences at 49 weeks on that measure. However, we did see some directionally favorable differences. So I'm just going to walk through that data with you now. So the primary efficacy for the forced stair climb time was based on, and you'll see this on the following slides, you'll see this MMRM. which you see in the first box after the 4th stair climb time. This is a statistical analysis method that's called a mixed model for repeated measure. It's used to account for missing data. For example, if the subject lost the ability to climb 4 stairs during the study, this model would take into account those missing values. So in addition to looking at the 4 stair climb time by this MMRM method, we also performed sensitivity analysis to determine the robustness of this MMRM analysis and to show that it was accurately measuring these outcomes. Some of these sensitivity analyses included looking at changes in velocity, and we also looked at patients who were able to complete the study, and we also looked at patients who might have been outliers. But as I said earlier, none of the sensitivity analysis were statistically significant. So this MMRM, this change that we saw over the 49 weeks in the DOMA-treated group, showed that these patients had an increase in their 4th stair climb time of 8.28 seconds, and the placebo group had an increase on the 4th stair climb of 8 seconds. So in this case, when we look at the difference between Domagrozumab and placebo, we see that there was a 0.27 second difference, which actually favors placebo over Domagrozumab, and however, that change wasn't statistically significant. I may also just add here that at the baseline, when we looked at the differences between the two groups, both the Domagrozumab treated group and the placebo group were about 5 seconds at baseline on average. So we also looked at this 4th stair climb time compared to this Synergy natural history matched control group. And as I said, we matched this group of patients from the Synergy data set who were the same age, were on a background of glucocorticosteroids, and had a similar baseline 4th stair climb time. So what we had was our week 97 included 18 boys who had completed through week 97, compared to 77 boys from the Synergy data set. So what we saw was that over the 2 years, the sequence group 1, our DOMA group, had an increase on the 4th stair climb of 4.2 seconds, and the Synergy group had an increase of 3.4 seconds. Again, the difference, 0.82 seconds, actually favors the Synergy group over the DOMA-treated group, and the p-value wasn't significant. So when we looked at the 6-minute walk difference compared to placebo, it was the same MMRM that was used. And what we saw was that the DOMA-treated group lost 58 meters versus the placebo group that lost about 56, which are very similar, and there was really no change or no difference seen in those groups. And I just want to comment that at baseline, these groups were very similar. The DOMA group on average was performing the 6-minute walk distance at about 370 meters versus the placebo group that was performing this task at about 350 meters. So the North Star was favorable in the Domagrozumab group, but it didn't reach statistical significance at week 49. So what we see in this is that the North Star score decreased in the Domagrozumab group by 3.6 points versus in the placebo group who lost 5.2 points, with a difference of about 1.6 points. So one of the things we asked ourselves was, well, was this different? Although it wasn't clinically significant, did it reach a threshold of what would be considered minimally clinically important difference? And that's a threshold where we have to demonstrate that this type of change would make a difference as a therapeutic effect. And a minimal clinical important difference for the North Star is around 3 points, and what we saw was that we were below that. So while the change was favorable towards Domagrozumab, it didn't reach statistical significance, and it didn't reach the threshold that would be considered a minimal clinical importance difference. So we also looked at the 10-meter run walk compared to placebo at 49 weeks, and again, we didn't see a difference in this on this test either, and the groups were similar at baseline. So as I mentioned, we also looked at this pediatric outcomes data collection instrument called the PODSI, which is a questionnaire, again, that's completed by Parent Project. It has about 36 questions and performed at each visit when we had functional outcome testing performed. And what we saw was that we saw some scales within the PODSI that looked like they were changing. Those that changed the most included the upper extremity and physical function, the transfer of basic mobility, the pain and comfort scales, as well as the global functioning. And we had two scales that had smaller differences, including the happiness core scale and sports and physical functioning core scale. And the image below is just a figure of that upper extremity and physical functioning scale, where red is the Domagrozumab treated group, which shows it had a favorable difference over placebo at 49 weeks. Again, it was not statistically significantly different. I also wanted to touch on the thigh MRI results. We had the thigh MRI performed to evaluate changes on muscle volume, the quality, and we also wanted to look at fat fraction. So we were very interested in muscle volume because you will remember that myostatin's goal is to regulate muscle growth, and we thought blocking the action of that protein may lead to increased muscle volume, which is what was seen in the mice as well as in cynomolgus monkeys. We also know that the role of myostatin is to replace tissue with fibrotic tissue, so we were interested in trying to see if there was any improvement in the quality of muscle, which we could measure by T2 mapping. And then finally, we wanted to look at fat fraction, which is obviously a known a predictor of longitudinal changes on MRI that measure that corresponds to the progression of Duchenne. So what we found on the MRI was that all the endpoints favored treatment with Domagrozumab after 49 weeks. However, we only saw on the T2 mapping-related endpoints that we had statistical significance. So this next slide here is the figure of the T2 relaxation time. And what we saw again was that in our DOMA-treated group, we did see a statistically significant difference on this relaxation time. So the T2, again, is a measure that's looking at muscle inflammation, fat, and edema, but it's probably primarily driven by fat infiltration. So while we did see a statistical significant difference on this marker, it didn't correspond with any differences or measurable differences on the functional assessment. We also used DXA during the trial to provide information on bone, fat, and lean body mass, and whole body DXA was performed to do that. In the DMD natural history study, DXA showed an increase with percent lean mass with age. And in the Domagrozumab-treated patients, we saw an increase in lean body mass at 49 weeks, which was favorable, but was not significant. We also did additional analyses in these groups, which you can see by looking at the figure, which is actually a DXA of a Duchenne patient. And the lines on the screen actually show how we segmented looking at the appendicular skeleton, which is the arms and the legs, And what we did see was a statistically significant increase in lean mass when we just looked at the appendicular skeleton. So this next figure just shows this whole body change over time, and it was favorable but not statistically significant. So finally, I just wanted to touch on our results and data sharing plans. So currently, we are in the process of submitting our basic results into clinicaltrials.gov, and we're hopeful that those will be posted in the next few weeks, as well as to submitting them into the EU Clinical Trial Registry as well. So those data will be available for you to view. However, we recognize that some of these systems may be difficult to interpret. So on Pfizer.com, we will also be posting a clinical study report synopsis, and that should be available shortly. We are also have met with the investigators who participated in this trial. We're obviously very interested in the amount of data that we've collected on the study. Then we are working with them on publication plans, with our plans to have our first publication submitted in the second half of this year, with others to follow. So Pfizer has a policy to ensure that we share data, and by that I mean that we provide clinical trial access to regulators, researchers, and trial participants. So on Pfizer.com, you can find that returning data to clinical trial participants is a priority for Pfizer. They feel it's very important for participants to understand the research they took part in and to see the data that was gathered about their health to really understand more about the clinical trial. So there is a place on Pfizer.com where you can actually request access to your own personal data. However, we also do have a policy that we don't release the data until 2 years after our primary completion, so this data won't be available until next April. Finally, I just wanted to summarize what we talked about today. So, as I said, our primary study did not meet the primary efficacy endpoint, and we did review the totality of evidence, including secondary endpoints, which really didn't support a significant treatment effect. So our studies were never terminated for safety reasons, and we are still conducting more exploratory data analysis given the vast amount of data that we have available from this trial. So we do feel really strongly that this is a high-quality data set, and we feel it's very important to get this data out into the scientific community as quickly as possible, especially around things that we've learned, such as the Glutamate Dehydrogenase, and being able to use this as a potential liver injury marker in muscle disease, as well as to provide information about the cardiac troponin I elevations that we saw as part of the natural history for Duchenne. We also learned through our MRI and DXA imaging protocols that we can scale imaging in large multi-center trials. And we also now have a placebo data set that is available for us, as well as an imaging data set as well, to understand more about the natural history of Duchenne. And really, I'll just say the impact of having all of this information available has had an immediate impact on the DMD gene therapy program, I'd say especially since we have this placebo data set that can provide and inform us how to best design those future trials. So finally, I just want to end with a huge thank you for the past 5 years. We're grateful to the families and the boys who committed their time to this study, the investigators and staff who conducted this trial with us, and the advocacy group who connected us with the patients and families, and we really look forward in the next year to sharing more results with these trials with you. Thank you.
Thank you, Shannon, and thank you, Dr. Charnas. This was really a terrific review of the Domagrozumab study. We do have a few questions. I'm going to start off with mine, and then I'm going to go with the others. Over time, we've talked about the different steroid regimens and their potential impact or not on a given study. Can you comment on have you looked at the three sequences to understand what steroid regimen they were on, and did you see any differences if you've done that work?
So we're somewhat limited in what we can do with our steroid data. We did not require the boys to be on any specific regimen. However, they were on a stable dose. And what we found is about 60% of the boys were on a high-dose daily regimen. But because the study wasn't designed to answer those questions about a regimen, we are limited in that we aren't able to really analyze how regimens might be different between groups.
Okay, thank you. And were you checking CK levels along the way, and did you see any differences in CK levels?
We were checking CK, and we did not see any differences.
Okay, and can you speak to the post hoc analysis?
That analysis is ongoing. There are current plans to present that at a scientific meeting in October. And right now, I think that's all we're in a position to say.
Okay, great. So, yeah, sure. So, Dr. Charnas, another question that I have for you and Shannon is, as you saw these favorable trends, but not statistically significant, if you could make the rules today, do you think that is extending that study out by a year, especially with sequence 1 being on a consistent active compound throughout the 96 weeks, do you think, and I know it's hypothetical, right? If you were doing the study again, would you extend it out another year to see if the favorable differences would be reach statistical significance?
If I could answer questions like that, I wouldn't need to do clinical trials.
Yes, see? Yes, see there?
Yeah, I understand. It's an excellent question. I think what you're really asking is what is the optimal length of a clinical trial and how do you show it? Clearly, there is real-world data from other observations that the longer you go, the easier it is to see the impact of subtle changes that may occur in aggregate. I don't know that we would design a trial like that, but it's an interesting suggestion, and we'll carry that forward.
Yeah, I think in Duchenne, so I think you well have heard from families that no change, no negative changes, so stability is benefit, right, in our view, because I think we get up every single day and look at our sons and say, don't let anything change. So when you see a favorable trend, you say to yourself, would that trend increase over time? Would that stay favorable over time? So I guess it is a question, especially in a disease like Duchenne, where the progression is over time, cardiac 1 to 3%, all of those things hard to measure in terms of how long you can run a study and certainly how much commitment the families are willing to make for those number of years. So I thank you. I know it's a difficult question. I also just wanted to ask a question about the sequence 2, where in sequence 2, those children were on active compound 2040, et cetera, and then they were off on placebo for the next 48 weeks. Did you see a change did you match that group from its baseline on active drug, did you measure them from active drug to placebo, and was there a change in that individual subset?
We did look at each of the sequences over time to see what happened with sequence group 1 when they were on the drug for 2 years, sequence group 2 when they started on the drug, and then they went on to placebo, and we didn't see a treatment effect when they came off the drug, if that's what your question is, in year 2. And we also looked at sequence group 3, thinking that the boys who might be in sequence group 3 might be a year older, if you will, by the time they started to receive the drug, to see if that made a difference, and we didn't see a difference in that group either.
Okay. Thank you for that. And then, as you might imagine, in a study like this or any of the studies, there are very individual differences. And so one of the questions would be from families, will their individual data ever be available to them? And if so, when?
Yep. So Pfizer's policy is to share individual level data, and this can be found on Pfizer.com. However, there is a policy in place that we don't release the data until 2 years after the primary analysis, so that the request for that data wouldn't be able to be made until next April of 2020. But Pfizer does want individuals to see their data that was gathered on when they participated in the clinical trial.
Thanks, Shannon. So I just wanted to reiterate that. So what they would do is, in April of next year, go to their clinician and request their individual data, and then they would be able to see it. Is that right?
They can actually go into Pfizer.com and request it. There's actually a process right through our Pfizer.com website.
Okay. Thank you very much. Well, I think that, so there have been questions about decreasing in fibrosis, which I think you covered well in the T2 and fat fraction. So, and you'll be looking at the imaging data in more detail to see if there's subsets in that group that you mentioned there are different subsets in terms of rates of progression that you found looking at the forced air climb. So will you be also matching that up to other, such as fat fraction and T2?
Yeah, so we are continuing to look at the imaging data to learn more about it. I think, one of the issues is when you don't see a big functional treatment effect, you're somewhat limited in, saying that somebody was a responder. But we have more work, ongoing to look to see if there are differences, say where they were from baseline on the imaging versus where they ended up a year later, and, were any of those patients, more responders than not. So that is ongoing.
And so these are the data that you'll be presenting at the national meeting or the World Muscle Society in October?
I can't say that for sure. Those would be included. But I think we do have plans to look at those to see if there is something there, and obviously we would plan to publish information as we learn it.
Great. Thank you very much. And so final thoughts from Dr. Charnas and Shannon. First of all, thank you for doing the study. I think none of us want to see terminated studies ever, but I think the fact that you've committed these 5 years and we've committed them to you as well, we've learned a lot about Duchenne, and we're thank you for that. Are there final thoughts? I can't thank the families enough. I can't thank the clinicians enough for all they do, and to you as well. We know that this isn't easy work, and it's heartbreaking for all of us to see a study that's terminated. That said, you've provided a wealth of data, and certainly a wealth of data to the community. So I thought I'd ask just for final thoughts from each of you. Dr. Charnas, you want to start?
Thank you.
Sorry, Chief.
The most important is that we have heard people describe this as a failed study. It's not. A failed study is a study in which you didn't learn anything and you can't move forward. Quite the contrary. We got an outcome that was not anticipated or desired, and we are saddened by that. But we conducted a well-controlled trial. We learned a tremendous amount. We can bring that forward not only within Pfizer but to the community. And therefore, this was a successful trial, but it did not produce a therapeutic drug. And I think that the language is important.
I agree with you. Thank you. Shannon?
Yeah, I guess I just wanted to reiterate at the beginning when we started this that we when we entered this community, we were seeing that it was really difficult to measure, therapies in this community, and we really set out to try to answer this question definitively, and I'm proud that we were able to do that. I don't, think that it was, again, I'll say, totally, not happy with the outcome that we had, but what we were happy about was that we didn't leave any stone unturned in terms of trying to say if this drug worked or not. And, everyone here at Pfizer really feels strongly that we need to provide good, effective therapies for these boys and therapies that don't, have marginal effects. So we really need, robust drugs for these kids who are so urgently waiting. So I do feel like, we set out to accomplish, answering that question. So I'm happy about that.
Yes, and we are too. Shannon, Dr. Jarnes, and I know your whole team. This is really a wonderful experience for us and a great learning experience, and certainly not the outcome we wished for. But in turning over every rock that these boys deserve turned over and looked for, we appreciate all you've done. And so for all of us on the phone, thank you, and thank your colleagues at Pfizer. For those of you who've joined us today, thank you for taking an hour of your day and getting it to us to learn more about this drug and what we've learned going through that clinical trial process. So this is Pat Furlong, and we're signing off and wish you a good day. Thank you so much.